Well, thanks everyone for coming today. I'm excited to be part of the symposium. So I'm going to be talking to you today about the promises and perils of monitoring change in native bee communities. I'm going to start off with some of the perils and then end on an optimistic note of some of the promises. First peril is community metrics. And what community metrics do we choose, or how do we define a native bee community? There are a whole host of different metrics. One of the most common is species richness. One of the problems, however, is that a lot of species richness estimators and accumulation curves are really susceptible to effort and sample size. This is a nice paper by Mike Roswell and others looking at the number of individuals on the x-axis. Each color represents a different site. We have the lines, which are species accumulation curves, and the clouds of points, which are Chow-1 species estimates, richness estimates. And what we can see here is that depending on the number of individuals, the rankings of the sites in terms of species richness changes. We see those lines crossing. In addition to that, the species richness estimators, those clouds of points, on some of these sites just to continue to go up and up. What's going on there is that there's a lot of singletons that are happening as individuals are caught. And what this means is that the uncertainty for these species richness estimates are it increases quite a bit. And it really makes species richness estimators really problematic and at times potentially not very useful. So to deal with this, ecologists have used more robust methods of incorporating relative abundance. This is things like Shannon and Simpsons and things like that. But this brings up another problem in the sense that these weight common species by more, and we're often concerned about rare species from a conservation perspective. Finally, one of the other issues of community metrics is that they don't often take into account things like species identity. So you can have things like non-native species that are the same as a native species in terms of how these metrics are calculated. And that can be not what we're interested in from a conservation point of view. And so these metrics, community metrics, might hide some of the important processes. Another important peril, and I'm going to be really quick about this one because Terry's talking next, and there's a whole RCN in a couple of weeks, is this idea of taxonomy. So for one, I think a lot of us know that identifying bees is difficult, and there simply isn't very many individuals that can do it well. Secondly, a lot of groups are not well resolved, and this is at least as big of a problem. So we're working from things that haven't been resolved in 50, 60, 70, 80 or more years. And so we really don't know what species we're actually working with. A third peril is the idea of variability. So this is a hypothetical, a figure of a hypothetical species here. On the x-axis, we have years. On the y, we have abundance. The gray lines indicate abundances over different years. And what we see here is a lot of variability. Again, this is hypothetical, but we see this in a lot of bee communities, a lot of insect populations. And this makes it really difficult to detect trends, of course. This is kind of basic stats. But in addition to that, it can lead to this thing called a false baseline effect. And what that is that you basically, depending on when you start the study and when you stop the study, you can get different inferences. So the blue line here for this hypothetical species tells us that the species is not changing over a 30-year period, but you can see from these different red lines, depending on when the endpoints were, you can infer big declines or big increases just by the timing of it. And so the only way to deal with this actually is to really do a lot more long-term collecting across years. So, but there are some promises as well. And one of the promises I do really think is, in fact, taxonomy. I think a lot of ecologists and as conservation biologists really do value taxonomy and see this as an important aspect and need to support it more. But in addition to that, I think we want to make a plug for really doing the best we can on curating our specimens that allows us and taxonomists in the future to incorporate revisions and misidentifications and make a big plug here to support your local collection. One of the things I do hope is that in 100 years from now, there's a scientist up here that is talking about some sort of pattern that they were able to uncover because scientists of today did such a good job supporting collections. Another really big, and this maybe is the biggest promise, is this interest in pollinators is really booming. This is some work by Becky Barak. She surveyed over 100 land managers in the tallgrass prairie region and asked them what their most important objective was for habitat restoration. And what you can see here is that habitat for pollinators was the second most important objective for these land managers, showing how important they value pollinators. And we see this a lot in our lab. We have state agencies, for example, asking us how to incorporate bees in their wildlife action plan and which species to incorporate. Federal agencies want to understand how habitat management influences pollinators. And these are really vital questions that they want to use to really implement policy or make management decisions. And so these stakeholder questions are also our questions as well, but we can leverage these questions to build longer term data sets and hopefully be able to see how communities change over longer time periods through these collaborations. So to highlight, I think that we really need for community perspective is local long-term monitoring. We need to leverage our taxonomists and support taxonomic research, curate specimens for university collections, and then really collaborate with local, state, federal, and NGOs. And that'll hopefully allow us to conduct longer-term local community level monitoring and hopefully get at some of these questions of change over time. Thanks so much. I appreciate your time.